When this bacteria is infecting the plant, bacteria is transferring their gene to the plant. Along with that, their own gene, it is transferring the gene of our desired or our interest to the plant. And when it is transferred, when the bacteria start multiplying, when the bacteria is multiplying, the gene of our interest will multiply, and that gene will start giving their characteristics. So whatever the characteristics of gene of our interest was there, we have put it in agrobacterium, and agrobacterium is infected to the plant, and now plant is ready to show whatever the desired character we wanted from other organism. There is another method. Here in this method we have no need to use any organism. Here it is particle bombardment. It's a kind of syringe. With the help of that syringe, we are directly bombarding the gene. One can see the photograph where in the culture plate directly bombarding is occurring. By this method, one can get immediate transfer of the gene. This is one method, electroporation and direct DNA entry into protoplast. In this method, first DNA is introduced with the cell suspension or on the Petri plate. After putting here, one can give electric shock. Mild electric pulses is generated. Because of electric pulses, there will be little bit change in the membrane of the cell. See, here this is normal membrane, but once the electrical pulses is generated, it will show a kind of pores. Permeability will be changed, and changing in this permeability will immediately help in entry of the DNA. One can say direct entry of the DNA, and after pulsing immediately normal condition will be there, but after pulsing DNA will be absorbed. Here, once the DNA is introduced inside the cell, the new cell will be having the DNA of our interest. By that way, we can directly transfer the gene or DNA, whatever you say. Genetic engineering is art. It's art for plant protection also. There are a lot many diseases, there are a lot many stress conditions, there are a lot many environmental conditions. From that environmental condition or the adverse effect, we can save the plant, we can protect the plant. So here, genetic engineering is ready to help there also. If you want transgenic plant which has resistance to insect, to virus, to fungus and to bacteria, genetic engineering is ready. Genetic engineering is ready to produce the plant which is transgenic and having such kind of resistance to the disease, to the environmental condition. So plant has no need to worry from any kind of problem. Let me see first insect resistance. BT is one of the well-known cultivar, BT cotton, which is from Bacillus thuringiensis. Here this protein is very helpful. That protein is toxic to certain insects, and because it is toxic to certain insect, the gene which is responsible for that protein is transferred to the cotton. Once the insect is feeding it, automatically the gene has produced the protein, and that protein will be harmful to the insect, and insect will die, but there will be no any harm to the plant in particular. There are certain protease inhibitors, amylase inhibitors, there are certain lecithins. This all are the material which is harmful to the insect, but it is not harmful to the plant, and automatically with the help of genetic engineering we can produce it, we can express it, and we can get resistance to the insects. For the virus resistance, we can have same method. If we can express the protein which is responsible for viral coat, which is responsible for replicase enzyme, which is antisense messenger RNA for viral coat protein, or which is messenger RNA for satellite RNA of virus, or for defective viral RNA, or for ribozymes. These are the certain genes which produce certain proteins, and that proteins will stop viral infection. Of course, talking is very easy. Till today, lot of effort has been done, but success ratio is not 100%. Some variety is there in the market which is having such kind of resistance, not all. Let me go through another one, that is the fungal residence. Fungal cell is having cell wall made up of chitin. That chitin can be destroyed with the help of enzyme called chitinase, and chitinase enzyme can be produced by gene. So if we transfer the gene responsible for chitinase production, we can get immediately destroy of the fungi. There are certain proteins which is inactivating ribosomes of fungi particular. So that ribosome inactivating protein can be expressed with the help of genetic engineering, and that will deactivate the ribosomes of particular organism and will kill that organism. Likewise, stimble synthesis, osmotin, pathogenesis related genes can be produced and RNAs can be produced. Same method can be used for bacterial resistance for saccharopin, lysozymes, threonine, toxin detoxifying enzymes and toxin resistance enzymes. If we summarize everything, what we have to do is, first we have to select. First we have to go for selection in the earlier method of plant breeding. After selection, we can go for hybridization, and after hybridization we can get new varieties. But in recent trends and the recently this genetic engineering era, we can have molecular biology and we can have biotechnology. In the earlier days, selection, hybridization and everything was done in the field and lot of time was consumed. Months and months was spent just to get new variety. Years and years were spent to get established variety. But now with the help of molecular biology, with the help of biotechnology, one can get it immediately within 1 year or within 2 years. We can get result. We can immediately get new variety which is having good resistance, which is having good characteristics and desired character of our interest. With this, thank you very much.